Good morning, dear students. My name is Anuja Kurevaru. Today we'll be seeing a technique that is PCR. Hope the slide is visible to everyone. Oh. Now see what is PCR. PCR is nothing but polymerase chain reaction. PCR is an in vitro technique for the amplification of a region of DNA which lies between two regions known as known sequences. PCR amplification is achieved by using oligonucleotide primers. These are typically short, single-stranded oligonucleotides which are complementary to the outer regions of known sequences. The oligonucleotides serve as primers for DNA polymerase, and the denatured strand of the large DNA fragments serve as the template. This results in the synthesis of new DNA strands which are complementary to the parent template strands. These new strands have defined 5' ends, the 5' ends of the oligonucleotide primers, whereas the 3' ends are potentially ambiguous in the length. Now we can see how in each cycle the amplification is carried out. First of all, you can see the template DNA carrying your gene of interest or the gene which you want. In the first cycle, you are inserting the primers. In every cycle, there is presence of primers, as whenever you are loading the sample, you are putting or you are inserting or you are adding the primers required for it. Now, in the first cycle, there will be the copy of two strands of the DNA into two different strands. That is, the copies of the gene will be 4. In the next cycle, there will be 8 copies. In the next, there will be 16 copies. In the next, that is 4th cycle, there will be 32 copies. While in the last 35th cycle, there will be 68 billion copies of the desired gene. Now, polymerase chain reaction. Each cycle of the polymerase chain reaction has 3 different steps. First is denaturation, second is annealing, and third is the extension. In denaturation, the denaturation of double-stranded DNA occurs at the 94 degrees Celsius for 1 minute. The denaturation temperature and the time depends on the GC content of the DNA, as guanine and cytosine are paired together with the 3 hydrogen bonds. That's why they require high energy and high time for the separation. Hence, for that purpose, the temperature as well as the minute or the time frame is dependent on the GC content. Now moving to the next step, annealing. To anneal means to attach. Forward and reverse primers, which are complementary to the 5' and 3' end of our gene of interest or the gene we want, they are annealed, that is added at the 54 degrees Celsius for 45 seconds. Now in this step, all the primers will bind to the target DNA. And last step is the extension step. So only DNTPs are added with the help of DNA polymerases from the primers. Like DNA polymerases binds to the primer and it recognizes the DNTPs which are having Mg2 plus onto them, and it attaches the DNTPs to the whole DNA, and then you will get a new copy of DNA. Your targeted DNA will be copied at each cycle. See, each cycle, denaturation, annealing and extension. Now the PCR reaction will contain your target DNA, 2 primers, that is forward primer and reverse primer. Thermostable DNA polymerase generally The most widely commonly used DNA polymerase is stacked DNA polymerase, then oligonucleotide or DNTPs, and Mg2 plus cofactor for DNA polymerase. Primer selection. How we'll select the primer? Primer is an oligonucleotide sequence which will target a specific sequence of opposite base pairing, that is adenine to thymine, guanine to cytosine of single-stranded nucleic acid. For example, adenine will pair with thymine and guanine will pair with cytosine in any given sequence. Now we'll see DNA polymerase. DNA polymerase is the enzyme responsible for copying the sequence starting at the primer from the single strand. Commonly used, stack an enzyme from the hyperthermophilic organism that it can sustain at hypertemperature, that is thermus aquaticus, isolated first at the thermal spring in Yellowstone National Park, USA. This enzyme is heat tolerant. It is useful in thermally tolerant, that it survives the melting temperature of DNA denaturation, which also means the process is more specific. Higher temperature results in less mismatches and more specific replication. Now we'll see this RFLP and electrophoresis in the next lecture. Thank you so much.